Hi. Um, oh, okay.
Aha. Welcome, everyone. So it is a very great pleasure for me to introduce our first keynote speaker of the conference. Daniela Witten is a professor at the University of Washington who I first became familiar with a number of years ago after reading and using a textbook that she was a co-author on called Introduction to Statistical Learning, which is an outstanding introduction to machine learning with a focus on statistics. And before and since then, she's had a truly illustrious career with more recognitions and awards than I could possibly mention here. And during all of that time, she continues producing methods that are statistically innovative for the analysis of high dimensional data, and additionally come with very usable and practical software. So I've been a big fan of Daniela Witten for a lot of years now, and am thrilled to have her here with us, even though I can't, yes, she is here, in fact, with the speaker. So if I can manage to do this, I'm going to send myself off stage and hand the microphone over to Daniela Witten. Thank you so much for joining us.
Thank you so much for the very kind introduction. I'm really excited to be here today. I hope you guys can hear me. I have to say this is a really special occasion for me because it's such an honor to be able to give a keynote at the Bioconductor conference, but also because I've never given a talk where there's like floating emojis flying past my view of the screen before. So this is really special. So thanks everyone for that. I feel sad because I'm about to go into full screen mode and I won't be able to see the emojis anymore, but I'll just have to imagine that you guys are going to still be laughing at my jokes very politely. Okay, great. Well, today I'm excited to talk to you guys about my work on selective inference for trees. I hope that you can see the screen. I'm new to using AirMeet. I'm sure that Levi will let me know if you can't. So the work I'm going to talk about today is called Beyond Sample Splitting, Selective Inference on Trees. And the idea here is that in reality, as many of you know, or many of us know, there's often a difference between how we tell people to analyze their data and then how people really analyze their data. And it's this difference between the theory and the practice that I want to really get at today within a particular context, which has to do with selective inference. So the idea is that in theory, we know what we should do with our data. We all know that before we see our data, we should actually have a list of pre-specified hypotheses that we plan to test. And then we should only ever test those hypotheses. And this is what we write in our textbooks. This is what we teach our students in Stat 101. And actually, we don't stop with Stat 101 teaching this to our students. This is what we teach our students all the way through. And this is what we need in order, for example, for our type 1 errors guarantees to hold for our hypothesis tests and so on. So basically, classical statistics really breaks down if this doesn't hold. In other words, if we don't have pre-specified hypotheses in mind before we see our data. But of course, in practice, the reality is really far from the theory. So in practice, most people, statisticians included, despite what we teach in our classes, we tend to visualize, cluster, perform dimension reduction, and more in order to explore our data. And then after this exploratory data analysis, we decide what hypotheses we want to test. So the question is, what is going on here? Does it matter that there's a disconnect between the theory and the practice? And to the extent that it matters, is this something that we can fix in a practical setting? So today I'm specifically going to talk about what happens if you have data that you wish to cluster. And I'm going to talk about this idea that I'm going to refer to as double dipping. So here we have around 100 observations measured on 2 features. And we cluster the data, cluster those observations using our favorite clustering method, where here I'm showing you a dendrogram from hierarchical clustering, although it doesn't really matter what type of clustering you use. Here I've clustered the data to obtain 3 clusters shown here. And now we might want to know, is there a difference between the means of the 2 clusters? So this is a question that comes up, for example, in the context of single-cell RNA sequencing data. where we cluster the observations. In that case, the observations are cells for which we have single cell sequencing data, single cell sequencing measurements. And then we want to know, are there subtypes among these cells? So we cluster the cells, and then we want to know, is the mean in the orange group different from, let's say, the mean in the green group? And I'll talk more about that application in a bit. But more generally, we're just going to be thinking about the idea where we've clustered our data, and then we want to test a hypothesis about those clusters that we've obtained. So in a little more detail, this is simulated data. So the first thing I did was I sampled 100 observations from just a noise model. So there's no signal in this data. This is just normal 0-1 observations. And then I cluster the observations because, of course, regardless of whether or not there's signal in the data, I can always perform clustering, and I'll always get some clusters, even if I've just clustered the noise. So I cluster the observations. And now what I want to do is I want to test for a difference in means between those estimated clusters. So I want to test to see, like, is there a difference between the mean in the green cluster versus the orange cluster, a difference in the mean between the green cluster and the purple cluster, and so on. And basically to do this, I just compute like the pairwise distances between each pair of cluster centroids. I divide by an appropriate denominator, which is just a function of the standard deviation. And what I find, if I just compute the standard statistical test, is that all three p-values are tiny. So in particular, in this example, they're all less than 10 to the negative 6. So if I were just to go through this workflow where I cluster data and I test for a difference in means using a standard statistical test, I would reject the null hypothesis resoundingly, even though in this data set, I sampled my 100 observations from a noise model. So I know that there's no quote unquote true clusters. But the problem here is somehow, because I conducted a hypothesis test about clusters that were estimated from the data, I've kind of goofed. And I'm referring to this as double dipping, because in step 2, I dip into my data to cluster the observations. And then in step 3, I use the same data to compute p-values for a difference in means. And that has cost me my type 1 error control. So just to be completely crystal clear, in this example, because all of my data comes from just a normal 0-1 noise distribution, I would like to see p-values that are just distributed uniform 0-1. And of course, that's not the case here. These p-values are 10 to the negative 6. They are not drawn from a uniform 0-1 distribution. Okay, so very often when we encounter these double dipping type problems in statistics, really whenever we encounter any problems, we have like this hammer that we often can really use very effectively, and the hammer is sample splitting. So we can try to solve this problem using sample splitting. So in particular, on the left, I'm showing just again, like normal 0-1 data with no signal, but now I split it into a training set in the middle and a test set shown on the right. And now I'm gonna cluster just the training data. And I'm going to apply those clusters that I got from the training data onto the test set. So there's a number of ways that I could apply those cluster labels from the training set onto the test set. The details don't really matter, but for purposes of illustration here, I used 3 nearest neighbors classification, where each test set observation was colored orange if the majority of its nearest neighbors in the training set were orange. And in the other case, the test set observation was colored green if the majority of its 3 training set neighbors were green. And so now we can say, okay, well, I now got these clusters not from the training data, but actually from the test data. So now I can test the null hypothesis that there's no difference in mean between the orange and the green observations on the test data. But once again, something has gone wrong. My p-value is now like 10 to the negative 5th. Once again, that does not seem like it's probably drawn from a uniform 0-1 distribution. And when I first saw this result, I was actually really confused because I'd been talking with a grad student who's now faculty about this problem, and I was pretty sure that sample splitting, again, would just be like a hammer that would be able to hammer at this nail. Sample splitting isn't usually the best solution, but it's usually an okay solution. And she showed me this result, and I was pretty confused. But then what I came to realize is like, the issue here is that when you're applying those training set labels to the test data, you actually are using the test set features to determine if each test set observation is orange or green. So even though you're not doing the clustering on the test data, you are in a sense double dipping, because the process of deciding whether a test observation becomes orange or green uses the actual data for the test set. So I'd love to chat more about that during the Q&A if there's any questions, but I want to move on now in the interest of time. Okay, so the point is sample splitting is not our get out of jail free card today. It also leads to an inflated type 1 error rate. Okay, so you might wonder, have I just shown you some like pretty pathological examples here, or is this really a systematic problem? And the answer is this is a systematic problem. So here's a QQ plot showing the p-value quantiles that you get using this quote unquote naive approach, where we double dip into our data on the y-axis and the quantiles of a uniform 0-1 distribution on the right axis. The 45-degree line in red is where we would like our p-values to be if our type 1 error were controlled. And of course, our type 1 error isn't controlled. You can see that those p-values shown in black are just way too low, indicating that we have very, very, very bad control of the type 1 error, or really a lack of control of the type 1 error. So the p-values aren't uniform. And in fact, the selective type 1 error rate in this particular simulated example is around 97%. So we reject the null hypothesis 97% of the time instead of 5% of the time, which was the nominal level for this test. Okay, so now I'm going to talk for the rest of this talk about two particular instances of double dipping in clustering and double dipping in regression trees. And first, I'm going to spend the majority of this talk talking about double dipping in the context of clustering. And then I'll quickly cover double dipping in regression trees. And what you'll see is that really the same ideas that allow us to solve this double dipping problem in clustering can also be directly applied for regression trees. Okay, so with no further ado, double dipping and clustering. So we've seen that this is the setup. We cluster our data, and then we want to ask the question, are the cluster means really different? Is there really a difference between the mean of the orange cluster and the mean of the green cluster, for example? So we need to formulate this a little bit more statistically. So we're going to let X be an n by q data set, and the ith row is drawn from a normal distribution with mean mu I and variance sigma squared times the identity, where sigma squared is known. The assumption here that the variance is sigma squared times the identity is not important. If you wanted your variance to be some capital sigma that is a p by p, or rather a q by q covariance matrix that isn't diagonal, then no problem. And in fact, in the application that I'll show later, my covariance will not be a multiple of the identity. But for the purpose of this talk, just the math comes out a little bit more simple if we just assume that the covariance is diagonal. And now what I'm going to do is I'm going to cluster the data to get C1 hat through CK hat. So these are the clusters that I get if I cluster the n rows of the data. So for example, C1 hat is the indices of the rows that are part of the first cluster, and C2 hat is the indices of the rows, the observations that are part of the second cluster, and so on. And I'm going to define mu bar CK hat to be the mean associated with the case cluster. So this is just we just take all the observations that are in that case cluster CK hat, and we just average their mu i's. So the first thing I want you to notice is that mu bar CK hat is a little bit weird. It kind of looks like a population quantity because it's a function of the mu i's, but it's also a function of CK hat, which are the estimated clusters from the data. So it's sort of a population quantity, but it's also sort of a sample quantity. And in fact, that's a problem that we're going to encounter in a minute when we try to test null hypothesis, which is that for some pair of clusters, k and k prime, we want to test whether the population mean of CK hat is equal to the population mean of C hat k prime. And the reason that this null hypothesis is weird and actually quite problematic is because in statistics, we test hypotheses associated with parameters. We don't test hypotheses about the data. And so this null hypothesis, again, it's kind of about a population parameter because it involves the mu i's, but it's also kind of about the data because CK hat is a function of X. So there's something weird and problematic about this null hypothesis. But if we just suspend disbelief for a minute and we just sort of naively try to just power through, not noticing the fact that this null hypothesis isn't really quite about a population parameter, then this leads us to a very simple p-value. This is the p-value for the quote-unquote naive test that I was showing earlier, where the p-value is just the probability under the null hypothesis of no difference between the cluster means of observing such a big difference between the cluster means by chance. So observing at least as big of a difference between the cluster means is what we actually observed. So the difference in cluster means that we actually observed is shown on the right. A random variable representing the result of a clustering difference in cluster means is shown on the left. And the denominators here are just chosen to make everything work out nicely. because with this particular choice of denominator, using the fact that the random variable x is drawn from a normal distribution, we have that this entire p-value is just the probability that a chi-squared random variable with q degrees of freedom exceeds some threshold. And of course, this is something that we know how to compute really easily. You can just look it up in R using just base R. However, as we saw, this naive p-value is not uniformly distributed under the null hypothesis. So in order to fix this problem, what we need to do is we need to account for the fact that the only reason that we're interested in this null hypothesis is because we happen to notice that it looked like C hat K and C hat K prime might be different clusters. Like when we clustered our data, we noticed that the observations in C hat K were quite different from the observations in C hat K prime. That's why we came up with those clusters in the first place. So instead of just saying what's the probability, assuming no true difference, of seeing such a big difference in cluster means, we actually need to ask what's the probability, assuming no true difference between the clusters, of seeing such a big difference in cluster means, given that clustering the data actually gave us those two clusters. So this is just in writing what I just said verbally. The key point is we need to limit ourselves to the universe of data sets that give us clusters C hat K and C hat K prime. Because if clustering the data hadn't given us C hat K and C hat K prime, we never would have been interested in this null hypothesis in the first place, right? There's a huge number of null hypotheses we might have considered, but we considered this particular one because our data sure did look like it had a difference between those cluster means. So in order to check whether that difference in cluster means is bigger than what we would have expected by chance, what we're going to do is we're going to, once again, condition on the universe of data sets that actually would have caused us to estimate these two clusters. And this p-value has a really nice property. It controls what's known as the selective type 1 error rate, which is the error rate given that the null hypothesis is true, but also given that we decide to test this null hypothesis. So the standard type 1 error rate that you learn about in your intro stat class is just the probability that we falsely reject the null hypothesis given that the null hypothesis is true. But the selective type 1 error rate is once again the probability that we falsely reject the null hypothesis given that the null hypothesis is true, but also given that we decided to test this null hypothesis in the first place. So this is the p-value that we're interested in, but unfortunately, just because I can write something down, and I've learned this the hard way, just because I can write something down doesn't mean that I can calculate it. And so in fact, this is a p-value that I don't know how to compute. The reason I don't know how to compute it is because the conditioning set on the right-hand side here, like the universe of data sets that would have given me these two clusters, I don't know how to write that out. That's just like not something that I can work with. It involves a lot of unknown parameters. So the idea here, and here we're borrowing ideas from the selective inference literature by Will Fithian and Jonathan Taylor and Jason Lee and others, is to condition on a bit of additional information. So rather than just conditioning on the fact that clustering the data gives C hat K and C hat K prime, let's condition on a bit more, where I'm gonna carefully choose the additional things that I condition on in order to make this computation tractable. And very importantly, it's okay for me to condition on more information in the sense that this will still allow me to maintain the selective type 1 error control. So I'll keep my selective type 1 error, which I really care about, but I'm going to have to select that more in such a way that it'll make it possible for me to compute this p-value. Okay, so what else do I want to condition on? And the answer is a whole bunch of linear algebra. So my p-value is going to be the probability of, by chance, seeing such a big difference between the cluster means, given that clustering the data gave me C hat K and C hat K prime, but also given a whole bunch of other stuff. And to be honest, you don't really need to look at this linear algebra because I'm going to show you what it amounts to in a picture in a couple of slides. So again, this linear algebra looks bizarre, but it is actually very intuitive. So please bear with me for a minute. But before we get there to that intuition, I just want to show you an important result. And the important result is that this p-value that I told you I'm going to calculate, which again is the probability under the null hypothesis of seeing such a big difference between the estimated cluster means, given that I estimated these clusters on the data and also given a whole bunch of linear algebra, is just the probability that a random variable phi exceeds some threshold, given that clustering x prime of phi gives me C hat K and C hat K prime. So again, this is like the first equality on this slide. The p-value I'm interested in is the probability that some random variable phi exceeds some threshold, given that clustering x prime of phi gives me the cluster CIK and CIK prime. Well, what is X prime of phi? Well, X prime of phi is a new data set, which is just a perturbation of my original data X. So the ith row of X prime of phi is equal to xi plus a constant if the ith observation is in the cluster CIK. It's equal to xi minus a constant if the ith observation is in the K prime cluster. And if the ith observation is in neither the K nor the K prime cluster, then xi prime phi's ith row is just equal to xi. Okay, so what does this look like? Well, here's my original data, X. In this little example, I have 3 clusters. The blue one I'm calling CIK, the orange one I'm calling CIK prime, and there's another cluster too, which is shown in purple. And the dashed line here represents the line passing through the blue and orange cluster centroids. And so on the right, I'm showing you X prime of phi, where phi is a large number. And so when phi is large, what X prime of phi is doing is it's just pulling CIK and CIK prime apart, while keeping the purple cluster, which is not the cluster of interest, where it is. So X prime of phi is just a perturbation of the original data that holds everything fixed except for the 2 clusters of interest, and it just pulls the 2 clusters of interest away from each other if phi is large. But if phi is small, it does the opposite. It keeps all the other clusters fixed while pushing the 2 clusters of interest towards each other. And if phi is equal to just the observed difference between the cluster means, then X prime of phi is just identical to the original data X. So the point is that X prime of phi is just a perturbation of my original data that just pulls apart or smooshes together the 2 clusters of interest while leaving everything else the same. Okay, so to summarize, we did all this weird linear algebra, and what this allowed us to do is to devise a p-value that amounts to just taking a perturbation of the data that either pulls apart or smooshes together the 2 clusters of interest, so that our p-value is equivalent to just the probability under the null hypothesis that a random variable phi exceeds some threshold, given that clustering this perturbation X prime of phi gives me my 2 clusters of interest. And so what we're saying in words here is that out of all data sets of the form X prime of phi that give me CIK and CIK prime, what's the probability, assuming no true difference in that means, of observing such a large difference in the sample means of CIK and CIK prime? So basically, all that linear algebra that I did a couple slides ago, it allowed me to consider not the universe of all possible data sets that give me CIK and CIK prime, but rather simply the universe of data sets of the form X prime of phi, which results from either smooshing or pulling apart the 2 clusters of interest. Okay, so this is the p-value that I'm interested in. I've told you that it controls the selective type 1 error. So now we need to think about how to compute it. And I've sort of hinted to you guys that this p-value is going to be something that I'm going to be able to compute because I added in that linear algebra in order to make this computationally tractable. So let's think about how we would compute this p-value. Well, it's just, again, it's the probability under the null hypothesis that a random variable phi exceeds some threshold, conditional on some set. So that just is a function of the CDF of a chi random variable with q degrees of freedom, truncated to a particular set, which I'll call S. And S is the set of phi such that clustering X prime of phi results in CIK, CIK prime. So this is something that I can compute in R. This is like one line of R code, very easy to compute, as long as I know the set S, as long as I can tell you that, like, for example, S is an interval from like 3 to 17.2. And this is a p-value that I can just immediately calculate in one line of R code. So we're almost done. We only have one question left. And the last question is, how do we compute the set S? Because again, if I know how to compute S, then I'm done here. So it turns out that computing S is actually the hard part. And so I'm going to now talk about how we can compute S. So everything that I've said so far in this talk applied to any type of clustering. I haven't so far said anything that's specific to, for example, hierarchical clustering as opposed to k-means clustering or spectral clustering or model-based clustering. But in order to compute the set S, we're really going to have to actually dig into the details of how a particular clustering method works. So starting now, the results that I'll show are for hierarchical clustering. Okay, so we have one really important result, which is actually written at the top of the slide. And the important result is that if you get C hat K and C hat K prime by clustering the data X, then there's only one way the clustering X prime of phi can also give you C hat K and C hat K prime. And the one way that can happen is if the trees are identical up to the n minus kth merge. So in other words, if clustering the data X gives you C hat K prime, and also clustering X prime of phi gives you C hat K prime, that's possible if and only if the dendrograms resulting from X and X prime of phi are completely the same until we get to the n minus kth merge in the tree. So what am I talking about here? Well, here's what I get if I hierarchically cluster some data X with 5 observations. I get 2 clusters. And the dashed line is representing the n minus second. So here, the 5 minus second merge in the tree. And on the right, I'm showing you what happens if I cluster X prime of phi. And so take a careful look. Notice that both of these dendrograms involve 1, 2, 4 in one cluster and 3, 5 in the other cluster. But notice also that these dendrograms aren't the same below the dashed line. And the reason they're not the same, well, they're not the same for a number of reasons, but an easy way to see it is that on the left, the first merge is between 1 and 4, and on the right, the first merge is between 2 and 4. And so the point of this result is that this cannot happen. It is completely impossible to have a situation where the clusters in X are the same as the clusters in X prime of phi, and yet there's a difference between the dendrograms below that dashed line. And the fact that this can't happen actually allows me to efficiently characterize the set of phi such that X and X prime of phi can have the same clusters. So in particular, the set of S such that clustering X prime of phi gives me C hat K and C hat K prime can be written as the intersection over the first n minus k merges in the dendrogram of the set of phi such that the same pair of clusters merge at step L in X prime of phi and X. Because the only way for the trees to be identical up to the n minus kth merge is if for each of those first n minus k merges, whatever merges in X prime of phi also has to merge in X. So we can take it a step further, and we can say that this means that the set S can be written as the intersection over every single pair of clusters that doesn't merge in the first n minus k steps in X. That's an intersection over order of n squared sets, where each of these sets is just the set of phi such that the dissimilarity between this pair of clusters in X prime of phi is greater than the maximum height at which any pair of clusters merge during the time that A and B are present in the dendrogram. So it's kind of a mouthful, but the key point is that actually, magically, this set here can be characterized as a quadratic inequality in phi for several commonly used types of linkage in hierarchical clustering. So if you're familiar with hierarchical clustering, you know that there's linkages involved that govern the way in which we compute the dissimilarity between two clusters. And 4 of the most commonly used types of linkage are average, centroid, median, and ward linkage. And for those types of linkage, we can actually show that this set is just the solution to a quadratic inequality, which can therefore be simply evaluated using just the quadratic formula, the quadratic equation. So in other words, to characterize the set S, we need to take an intersection over order n squared sets, where each intersection involves a set that can be computed just in constant time. So this means we can compute the set S in constant time for average, centroid, median, or ward linkage, which is really cool. And it turns out that we can actually also compute S in order of n squared time using single linkage, which is another commonly used type of clustering. Although the actual argument for single linkage is different and also simpler than the one shown here. Okay, so to review, we want to test this null hypothesis that the mean of two clusters equals the mean of one cluster equals the mean of another cluster. And we showed that a p-value that involves just the CDF of a chi distribution with q degrees of freedom will allow us to control the selective type 1 error, but it's actually a truncated chi distribution. It's truncated to a particular set, so we had to characterize the set. And we can compute that set in order of n squared time for average, centroid, median, ward, and single linkage. Okay, so at this point, my hierarchical clustering fans in the house are maybe wondering, well, what about complete linkage? Because complete linkage is like the remaining type of linkage that's very commonly used in hierarchical clustering. So if you notice that it's missing from the slide, then you're right. And it turns out that we do not have a way to compute the set S efficiently for complete linkage. Wish we did, but we don't. However, we do have an efficient recipe to approximate the p-value using importance sampling. So even though we cannot compute it analytically in order of n squared time for complete linkage, we can still do it pretty efficiently. And somehow it's not on this slide, but if you're interested in some other type of clustering, like k-means clustering or spectral clustering, you could actually also take our importance sampling approach, our Monte Carlo approach for approximating the p-value for complete linkage, and you could apply it to your favorite clustering method. So though we only have like the ability to compute a p-value in order of n squared time for these particular types of linkage for hierarchical clustering, if you're interested in this framework more generally, then you can apply it with an arbitrary type of clustering. And actually our R package, which I'll mention in a couple minutes, allows you to do that pretty easily. Okay, so just quickly to show you what happens under simulated data. Here we've simulated data with no clusters. We obtain 3 clusters, and we test the null hypothesis for a randomly chosen pair of clusters. We compute the p-value exactly for average centroid and single linkage, and then we approximate the p-value using importance sampling for complete linkage. And what we see, thank goodness, is that our type 1 error is perfectly controlled. So these 4 panels correspond to 4 different types of linkage. The different colored lines correspond to different details of the simulation setup. But what we see is that everything is perfectly on the 45-degree line, so type 1 error is controlled, which is great because it would be pretty uncomfortable if I made it to this point in the talk and I was like, hey guys, this doesn't work. So good. P-values look uniform. We've got selective type 1 error control. Okay, so now I want to show you an application to a Palmer penguins data set. This is a data set consisting of 3 types of penguins, chinstrap, gentoo, and Adelie penguins. And we subset the data to 165 female penguins for whom we have complete data on species, bill length, and flipper length. So what we did was we applied average linkage hierarchical clustering to get 6 clusters. And just to emphasize, this clustering did not make any use of information about the species labels. I happen to know the species of the penguins, but I didn't give that information to my clustering procedure. So the data here with respect to the 2 dimensions, bill length and flipper length, is shown. Each cluster is a different color. And so the first thing that I can do is I can think about cluster number 1 and cluster number 2. And I can ask the question, is there a true difference in means between these 2 estimated clusters? Or is the difference that we're seeing no more than we'd expect by chance, given that these clusters were defined based on the data? So I happen to know that most of these penguins are Adelie penguins. I can see that because it's mostly circles within these 2 clusters. So I might suspect that in fact that there's not a real difference between the means of these 2 clusters. And in fact, if I perform just a naive test that doesn't account for the fact that I double dipped in my data, then I get a very small p-value, less than 0.01. But my p-value that accounts for the fact that these clusters were estimated from the data is a very modest 0.87. Okay, now what if I think about these 2 clusters instead? I happen to know that cluster number 1 is almost exclusively Adelie penguins, and that cluster number 4 is exclusively gentoo penguins. So I might suspect that there is a true difference between these estimated clusters. And interestingly, in this scenario, both the naive p-value that doesn't account for double dipping and my p-value that successfully controls the selective type 1 error, they're both very tiny, on the order of 10 to the negative 15. So we would reject the null hypothesis here. So what's really cool about my selective p-value is that when there doesn't seem like there should be a true difference in means, we actually fail to reject. But when we think that there is a true difference in means, we tend to reject successfully and really to not lose any power relative to the naive approach. So it's pretty nice to not lose much in terms of power while also maintaining selective type 1 error control. Okay, so just briefly, an application to single-cell RNA sequencing data. So this is a diagram made by Stephanie Hicks. And I know many of you are very familiar with single-cell RNA sequencing data, so I won't even spend time trying to explain it, except to say that we took a data set from a paper published in 2017, massively parallel digital transcriptional profiling of single cells. And this data contained various cell types, T cells, B cells, and monocytes. So the first thing that we did was we took 600 T cells, and I'm going to refer to this as the no clusters data set, because we're going to cluster the 600 T cells, and I believe that these are all T cells. So at least in broad brush strokes, there should be no true clusters in this data. And then we also subsampled 200 B cells, 200 T cells, and 200 monocytes. I'm going to call this the clusters data set. We pre-processed the data, and then, as I mentioned earlier, the results that I've showed in this talk assumed that the covariance matrix was a multiple of the identity, but there's really no reason for that assumption other than to make the notation on my slides simpler. So the tests that we applied allowed for a full covariance matrix, which we estimated using an approach published a few years ago. Okay, so the first thing that we did was we looked at the quote-unquote no clusters data set, which consisted of a single cell type, and we applied word linkage hierarchical clustering to get 3 clusters. So what you're seeing here are those 3 clusters shown in green, red, and blue, but you're seeing the projection of the data onto the first 2 principal components. And as you can see, those first 2 principal components only explain around 5% of the variation in the data. So that's why it visually looks like there's not good separation between the clusters, but that's because this is very high dimensional data and you're only seeing 2 dimensions right now. But what we can see is that the naive p-values that don't account for the fact that these clusters were estimated from the data are nonetheless very small, like 0.001, whereas our p-values are quite modest. They're around 0.7. And so this conforms to the fact that we happen to know that these are all T cells. Now, by contrast, we can also cluster the data that resulted from taking 200 T cells, 200 monocytes, and 200 B cells. And once again, I'm just showing the projection of the data onto the first 2 principal components. Although, of course, we clustered the data using all of the dimensions. And what we see is that both the naive p-values and our p-values are around 0.001. And furthermore, our p-values are almost identical to those naive p-values. So once again, when we believe that when we have reason to believe that there is a true difference between the clusters, then we tend to not only be able to reject the null hypothesis, but also really to not lose much power at all relative to naive approach that doesn't control the selective type 1 error. So this is really nice and reassuring. All right, so finally, in just the 5 minutes that I have left, I want to talk about the second part of my talk, which, as you'll see, really follows directly from the first part, and this involves double dipping in decision trees. So in particular, I'm going to be talking about decision trees resulting from CART, which is the classification and regression tree algorithm from Leo Breiman and others, which is by far the most popular way to build a decision tree. So just to explain to you what the double dipping problem is, I'm sampling 100 observations, which are shown here, but now associated with each observation, there's a response Y, which is also just drawn from a noise model. And so the value of Y is represented by the color of the observations on the left, where a big value of Y is a light color, a really negative value of Y is a dark color. And then I'm going to fit a decision tree using the CART algorithm. And in this case, I'm just fitting a very small decision tree, which you can see on the bottom. It just has 2 terminal nodes. And this corresponds to really just looking for the place that I can cut the data, either vertically or horizontally, in order to make the difference in the mean of the terminal nodes as big as possible. So I could have put that red line on the top anywhere, either horizontally or vertically, but I put it where I did because that's the spot that sort of increases the separation between the mean of Y on one side versus the mean of Y on the other side of the data. So now we can think about testing the null hypothesis, that there's no difference between the means of these 2 terminal nodes. There's no difference in the mean between the left and the right. Of course, in the sample, there's a very big difference between the mean on the left and the mean on the right of this red line. And the reason there's a really big difference in the sample is because I literally could have put that red line anywhere, either horizontally or vertically, in order to make it so that there's a difference between those sample means. And so, of course, it's not surprising, given that that red line was placed based on the data, that it'll look like there's a difference between the sample means. So indeed, just a naive test that just tests for a difference in those sample means using, for example, a 2 sample z-test gets a very small p-value, even though I know that I just generated this data from a normal 0-1 noise model. And so there really isn't a true difference between those means. Okay, so to phrase it a little bit more statistically, first we need a little bit of more general notation for a tree. So a tree is just something that looks like this, where we're going to have this notion of sibling nodes. So in this diagram, RA and RB are siblings because they come out of the same parent node. We're going to consider a data set XY, where X is fixed, Y is drawn from a normal distribution with some known variance. I'm going to fit a cart tree to predict Y using X. And then for any region R, I'm going to define Y bar sub R to be just the mean of the observations in that region. And mu bar sub R is going to be the population mean of the observations in that region. But these regions are going to be defined based on the data from the cart tree. So this quantity mu bar R, once again, is a little bit weird because it involves the observations in R, and R is itself a function of the data because it was estimated using cart. So the question we're going to ask is, can we test the null hypothesis the mu bar RA equals mu bar RB? And again, in this diagram, that amounts to testing the null hypothesis that the mean of the observations in the sibling node RA equals the mean of the observations in the sibling node RB. I should say the mean of the responses. The mean of the Ys in RA equals the mean of the Ys in RB on a population level. And again, this is just a weird hypothesis to test because the null hypothesis itself is a function of the data by way of the terminal nodes RA and RB. Okay, so you won't be surprised to know that as in the case of clustering, the naive test doesn't work. If we just try to brute force this and just compute a p-value, which is the probability under the null hypothesis of observing at least this big of a difference between the means of the terminal nodes RA and RB, and we just use the fact that Y is drawn from a normal distribution, so Y bar RA minus Y bar RB is also drawn from a normal distribution, then that gives us a p-value that does not have a uniform 0-1 distribution under the null hypothesis. So to fix this, we're going to really just mirror what we did in the case of clustering. We're going to condition on the fact that RA and RB were obtained from a tree. So we're going to test the null hypothesis, or rather, we're going to compute the probability under the null hypothesis of observing such a big difference between the means in these two terminal nodes, given that RA and RB are actually siblings in a tree. And so basically, just to make it a long story very, very short, computing this exact p-value is something I don't know how to do because I don't know how to characterize this conditioning set, but we condition on a bit more information, sort of similar to what we did in hierarchical clustering. And that gives us a p-value that amounts to just the distribution of a random variable conditional on some set. And it turns out that we're able to efficiently characterize that set for trees grown and pruned using the CART algorithm as the intersection of a bunch of quadratic inequalities using some really clever tricks and new results about the CART algorithm. Okay, so just finally, I want to close with a couple of results here, or really one result. So we looked at a data set called the Box Lunch Study, which is a randomized trial of the effect of portion size intervention on the number of calories consumed by trial participants. And in the initial study, the investigators used CART to identify subgroups of patients who differ in terms of their baseline caloric intake based on a questionnaire and a lab-based assessment. So the CART tree that they showed in their paper looks something like this, where they found, for example, and I'm sorry that these ovals are covering up the labels, but they found, for example, that whether or not you're hungry affects, maybe not surprisingly, your caloric intake. That's the top of the tree. And then the amount that you want food also affects your caloric intake and so on. So what we're able to contribute here is we can now actually compute p-values corresponding to each of these splits on the tree, testing the null hypothesis that there really is a difference between the terminal nodes coming out of that split. And you can see our p-values circled in pink. And what we can see is that these p-values are not statistically significant. So for example, the p-value for whether there's a difference in caloric intake between people who reported hunger less than 1.8 versus hunger greater than 1.8, that p-value is 0.44. So while this tree may have value, for example, in the prediction context, it really calls into question the extent to which we should be interpreting these splits on this tree and interpreting the terminal nodes, given that we don't find any of these splits to be statistically significant using our selective inference approach. All right, so I just want to close by saying that I've talked about 2 papers today, both of which are on archive, linked through my website. We also have software that I would love for you to check out. Of course, our software is in R. We have GitHub pages with really, hopefully you'll find user-friendly instructions for applying our R packages. This is the software page for our clustering work, but there's a similar tutorial available for the decision tree work. And just to close, I want to thank my wonderful collaborators. So Anna Neufeld is a current UW statistics PhD student who really led the work in the CART part of my talk. Lucy is my former PhD student at UW Biostat, who's now faculty at University of Waterloo. She led the work on hierarchical clustering and was also a key player in the work on trees. And finally, my longtime collaborator, Jacob Bian at University of Southern California, was involved in the work for the first part of this talk on hierarchical clustering. So thank you so much to all of you for your attention. I would be very happy to take any questions.
Thank you so much, Daniela. And I just wanted to assure you that when you showed your p-value diagrams and said how it would be awkward if you got to this point of the talk and said it didn't work, I laughed out loud and saw a whole bunch of laughing. So I'm pretty sure
Thank you
people
so much.
were laughing at your joke. Someone just put the question actually up on stage. I don't know if you can see it, but the first question that we saw here is, did you just solve the problem of what is the true number of clusters in a data set?
Yeah, that's a great question, Wolfgang, and I wish that I had. But what we're asking here is actually it's really kind of a different question. It doesn't have to do with the notion of really what are the true clusters, which is kind of a harder question to answer and even really a harder question to ask because it so closely depends on what model you're thinking about. Here, we're just asking the question of like, okay, you estimated some clusters. Corresponding to the observations in this cluster and the observations in that cluster are some underlying population means. Are those population means the same or are they different? So this is a question that we can ask even if we've estimated the clusters completely wrong. Even if, for example, there might be true clusters in the data, but we've only captured like 40% of one cluster and 36% of another cluster. Or maybe there's true clusters, but the clusters we've estimated are kind of a mishmash of those true clusters. Our question still makes sense and it's still well formulated. And actually, I think the question that we're asking makes a lot more sense than wondering about what's the true number of clusters in a data set, in the sense that in order to be able to really engage intellectually with the question of what's the true number of clusters in the data set, you need to just like have a lot of faith that you really have the right model. Whereas here, what we're doing, this is like pretty model light. We have like this normal distribution assumption. But other than that, it's like, great, you had some procedure that estimated some clusters. Now I just want to know, did you find signal or noise? I'm not worrying about if it was the right number of clusters or anything like that. So to answer your question, we solved a less ambitious, but I think much more practical question.
I had a question myself. I've been trying to think from the start of the talk about whether I've had some wrong intuition about test sets in the context of clustering and classification for a while. So I just wanted to check, if someone hands me a completely specified classification model and I use that to make classes in my data and then calculate a difference in means on those, am I still double dipping?
So somebody hands you like labels? Is that what you mean?
No, just a completely specified model. Let's say the CART tree, but they've already given me the decision points. So I'm not clustering in my data.
Did they get those decision points from your data, or did they get it from a different data set?
A different data set. And maybe an example relevant to this audience would be that there's a popular single R package that comes with its own reference cell data sets that it uses to develop the classifier on. So it does not use your data set.
So I think we need to be a little bit careful about what is the exact thing that we're doing. Because going to the example of CART, like if you give me a CART tree and you're like, here's a CART tree that I developed on my data, does there seem to be a difference between the terminal nodes for this cart tree using your data, then that's fine. That is a problem where like it's sort of similar to sample splitting. Sample splitting works okay for cart. But for clustering, it's a little bit more subtle, and I think we'd have to dig into the details of exactly how you're using that like pre-specified clustering that someone gave you. And the fact that sample splitting doesn't work for clustering, I was like super confused when I saw that for the first time. But I think the best way to think about it is just like imagine, it isn't even really about clustering. It's a more basic thing. Imagine you just have like a ton of marbles and you just drop them onto the floor. And then you have like a ruler. This seems like a very weirdly specific example, but this is just how I think about it. You have a ruler and you then drop the ruler on the floor. And then you see which marbles are on one side of the ruler, which marbles are on the other side of the ruler. You didn't have any intelligent strategy for placing that ruler. You just dropped it and it separated the marbles in some way. But then if you do a test for are the marbles on one side of the ruler in a different place from the marbles on the other side of the ruler, of course they are. Because the way that you separated those marbles was based on the ruler. So it's actually not even anything specific to clustering. It's just by virtue of the fact that you're deciding what null hypothesis to test by looking at the positions of those marbles that you've accidentally dipped your data, even if the way that you get those even if your clustering algorithm is as stupid as just dropping a ruler on the ground. So basically, yeah, I'd love to know more about the specific example you're thinking about with clustering. I feel confident that with cart, like you're fine if someone gives you a cart tree and you just want to test it on your data, but I'm not quite so sure about clustering, and it'll depend on the details.
All right, we have a bunch of questions. So I should ask some of other folks' questions. Can you give us an intuition about why you need hierarchical clustering to get the exact p-value, and why is it more difficult to compute the set S for partitioning algorithms from David Ariso?
Yeah, that's a great question. So as you're noting, Davide, our hierarchical cluster or our exact p-values are computed using hierarchical clustering. But I think that you could, for many other types of clustering, you could also take a similar approach to get exact p-values. Like I have a grad student who's interested in developing a similar framework, but using k-means clustering. So it's not that it had to be hierarchical clustering, it's just that that's what we wrote the paper about. But the point is, like characterizing that set S involves the intricacies of some clustering algorithms. So just because you've been able to characterize S for hierarchical clustering, that doesn't mean that immediately you can do it for k-means. You'll have to kind of like start from scratch to figure out what is the k-means clustering algorithm actually doing, and how can you characterize that in terms of the set phi. So I'm optimistic that could also be done for other types of clustering, but hierarchical clustering is just where we started because, first of all, because it's a really popular type of clustering, but also because we were able to sort of see how to do it.
All right, we have another question here. Could you comment on whether you would expect to run into the same type 1 error rate if you use independent subsets of genes for clustering versus mean calculation?
Yeah, that's a great question. So I think that what this question is asking is like, okay, well, let's say you want to cluster your cells based on, let's say, single cell RNA sequencing data. And so you just split your genes into two and you cluster using half the genes. And then you evaluate those clusters using the other half of the genes. So I think that the problem here is that genes are really dependent, right? So like if you kind of had some scenario where you had two sets of genes that were totally independent from each other, then I think, and this is just like off the cuff, and I certainly might be wrong, but off the cuff, I think that you'd be okay. But the problem is that there's, of course, like very high dependence between genes. And even if you have like quote unquote like gene modules or whatever that you feel in your heart might be kind of independent-ish, I would be quite worried. But that would be an interesting thing to check.
How do we convince people doing a single cell RNA-seq to stop double dipping and to use this approach?
I mean, well, just to be completely clear, like, you know, a lot of the analyses that people are doing with single cell RNA sequencing or in any practical setting are much more sophisticated than what I've shown here. So like if the exact thing that you're doing is hierarchical clustering, then like great, I have like an alternative procedure that you can use. But very often the things that people are doing are kind of like even more complicated and even more sophisticated. So I think that the first thing as a statistician that we can do is like kind of recognize the problem and start to characterize the ways in which the problem is arising and fixes for some of those problems, but I think it's a long process to like develop like a full framework that'll allow people to fully avoid double dipping on their data. But I think that in the interest of like scientific, replicability, in the interest of just like having results in our research studies that we understand, we should at least get people to understand the issues with double dipping, if not being able to give like a full end-to-end solution, because we're definitely not at the point of an end-to-end solution right now.
Thank you. And I think, oh, here's one more we can put up. Is there a good formal definition of double dipping?
Yeah, I mean, double dipping is just like the word that I like to use, because for me, it really evokes the problem. But I think the more formal way to think about this has to do with the selective type 1 error. So like, when you run a test, are your p-values going to be uniformly distributed when the null hypothesis doesn't hold, given that you decided to conduct this test? And if your p-values are uniformly distributed, then you're fine, there's no problem. But if your p-values aren't uniformly distributed, and typically they'll tend to be anti-conservative, which means too small, then there's an issue here, because all of your findings are going to be problematic. So I guess to answer your question, I would say generating QQ plots for your particular statistical test is a fine way to think about it. I also just want to say, I've given this talk before, and there's always someone who's like, oh, but like p-values are so like 2000s, and now we just use confidence intervals or whatever, and it doesn't matter. The same ideas that I'm talking about here apply just as well to confidence intervals. It's just that p-values are kind of more convenient to talk about. But as you know, there's a mapping between p-values and confidence intervals. So if your p-values don't work, neither do your confidence intervals.
Thank you. And the last few questions that I had were all about links to your slides and to the software. I believe the software link I have here, but
the software is available. I see that someone posted a link to the cluster p-value software, but if you look on my website, which is danielawitten.com, then there's also a link to the decision tree software. And our papers are there. The papers are linked.
And if you do want to share a link to your slides, we can attach it to the session here for people.
Oh, great! Thank you.
Well, thank you so much, Professor Witten. It's been really an honor to host you here. And although it's hard to tell when you're online, we've had between 200 and 250 people watching, so it's not a bad auditorium of people, and a lot of appreciation, I know. So
well, thank you so much. This is such an honor to have the chance to speak here today, and thanks to everyone for the questions and to you for hosting the session.
Thank you.